I will now introduce our next speaker, Professor Clare Scott. She will be speaking to us about precision in targeting rare gynecological cancers, streamlining where possible, and diving deep. Professor Clare Scott holds the chair in gynecological cancer at the University of Melbourne and is Joint Division Head of Clinical Translation and Laboratory Head at the Walter and Eliza Hall Institute of Medical Research and Medical Oncologist at the Peter MacCallum Cancer Centre at the Royal Women's and Royal Melbourne Hospitals. She has 25 years of experience in clinical cancer genetics, and her lab primarily focuses on drug resistance in ovarian cancer and other rare cancer types. Professor Scott also chairs the COSA Rare Cancer Group and the Board of International Rare Cancer Initiative, and has been awarded clinical fellowships from several international organizations, and she's also a fellow of the Academy here. Claire, if you can, hope this all works, and start your talk. Thank you very much.
Thank you. It's a very great pleasure to be asked to give this talk. I'm extremely honored, as are the other speakers. So I'd like to discuss precision medicine in targeting rare gynecological cancers. We've attempted to streamline wherever possible and to dive deep in order to gain additional insights. As you all know, outcomes are improved in medicine by evidence-based medicine, but many rare cancer patients do not have evidence-based treatments or access to research. So in order to interrupt this cycle, we're involving as many rare cancer patients as possible in research and providing flexible research platforms for them. And I'm going to describe a number of opportunities which we've generated in order to try to access drugs for patients. I'd like to take this opportunity to give a shout out to Kate and Richard Vines, who've done more than anyone else in Australia to really upend medicine and government and pharma in this end. So just very quickly, we take the Gatter et al. Definition of rare cancers, which is an incidence of less than 6 per 100,000 population per annum. And collectively, this accounts for 22% of all cancer diagnoses, but most importantly, for a third of all cancer deaths. So these Australians are dying of cancer in the main without access to evidence-based guidelines, and certainly usually without access to modern treatments or targeted treatments. Their outcomes are inferior to those with common cancers, and often the precise cell of origin and etiology is unknown, which means they're being given treatments just according to the closest histopathological or tumour stream guidelines. I'm going to talk about ovarian cancer because it provides a very good example of gynecologic rare cancers. The survival rates have changed little over the last 30 years, and we hope that that is changing, and I'm going to describe why. Poor survival rates in ovarian cancer are largely due to the fact that disease screening and early detection is lacking, generalised approaches to treatment are used, and there have been inadequate preclinical models. I'm going to describe high-grade serous ovarian cancer molecular subsets. Up to 50% of high-grade serous ovarian cancer are known to have defects in HR pathways, which I'll describe. And BRCA1 and BRCA2 mutations are usually quite well known and account for a significant subset. So PARP inhibitors are the most exciting treatment for ovarian cancer, and I'll show you why shortly. The homologous recombination DNA repair pathway is very important and underlies susceptibility to PARP inhibitor therapy because of so-called synthetic lethality. PARP inhibitors cause PARP trapping onto DNA and blocking of progression of replication forks. The SOLO-1 trial is a trial of women with BRCA1 and 2 germline mutations who received 2 years of PARP inhibitor therapy in the first-line maintenance setting following chemotherapy. And last month at ESMO, the 5-year update was provided, really showing that for some patients, there is an increased chance of cure, which in solid cancers is unprecedented. I'll just show you one piece of data showing that nearly 50% of women remain disease-free at 5 years after only 2 years of therapy. Now, in my lab, we've been making patient-derived xenograft models, and this really enables molecular dissection. We get very pure tumor concentration, and we're able to ask really rigorous molecular mechanism of action questions. So we have molecularly annotated cohorts of different rare cancers, and in particular, high-grade serous ovarian cancer, PDX with rare genetic defects. And I'm going to talk about a number of these. I'm going to look at distinct mechanisms of therapeutic resistance today, in particular RAD51C and D mutation reversion and methylation reversion. So here we're going to look at one PDX which had RAD51C secondary mutation. So a patient of mine had been responding to a PARP inhibitor on a clinical trial for about 9 months when she recurred. And in that recurrence, we discovered that she had a previously unknown RAD51C mutation. And in her recurrence, there were multiple, up to 4 secondary mutations. We found that we could look at these in great detail in the PDX, and we then manifested those in a cell line and made several unique discoveries. So in the TCGA data, the BRCA1 methylation had not been shown to confer survival advantage. So in our PDX, we were able to show that there was a mixed response in the BRCA1 methylated PDXs, and we had some PDXs responding and not others. However, in our PDX, with help from our collaborator, Alex Dobrovich, we were able to show that some PDX had really perfect methylation of BRCA1, and in other PDX, we could see that there was only heterozygous or half an amount of methylation. So we were able to study this with 3 types of analyses of methylation, and also look at the relative expression of BRCA1 to show that indeed in our homozygous methylated cases, there was no expression, and in heterozygous, we had half an amount of methylation. We then went to an associated clinical trial where we were able to show that cases where there was homozygous methylation, we had really neat responses, but in cases with heterozygous methylation, they overlaid wild type cases. So this showed for the first time that BRCA1 methylation could be used to predict PARP responses. And methylation zygosity as a concept was indeed similar to mutation zygosity for BRCA1. This concept had never even been considered before. BRCA1 methylation loss could therefore happen after exposure to chemotherapy, and loss of methylation of just one BRCA1 copy was enough to cause resistance. This needed to be validated in more patients, particularly in the early treatment setting, and this is incredibly important given the SOLO-1 data, because just recently in the federal budget, our PBAC have approved first-line BRCA1 maintenance therapy for BRCA1 mutation carriers, but of course not methylation of BRCA1 women. And that means these women who critically need it before this BRCA1 methylation is lost are still not able to access this drug despite their rare cancer. What about RAD51C methylation? So in our PDX183, we knew that there was a PARP response, but no HR mutation present, but indeed we were able to show that there was no RAD51C expression. We did find, however, that there was RAD51C methylation. We also had another PDX case and indeed 12 patients by trolling various cohorts. It takes a long time to identify these rare cases. And indeed, by a number of mechanisms, we were able to show a unique pattern. So a new pattern of RAD51C methylation not previously identified, but we identified the same rules that we'd shown for BRCA1 methylation. So having described this new pattern, we were able to show that homozygous methylation also predicts PARP response, that it can be lost under PARP pressure. And now we have an EMBRACE clinical trial here in Australia of olaparib at the NHMRC CTC, and the PI is Katrina Soquist. And in that trial, in addition to other rare indications for olaparib, we are able to further validate the BRCA1 or RAD51C methylated breast and ovarian cancer cases to better validate these findings. Now, what about a wider search for rare cancers. So this nominated trial, genomic matching treatment for rare cancers, was first initiated by discussions with Kate and Richard Vines, our rare cancer consumers. They wanted a nationwide study so that rare cancer patients could access molecular profiling. And when we started this, that was our aim, just to determine whether rare cancer cases could be molecularly profiled and we could establish that there were aberrations suitable for treatment, because in those days that was not at all accepted by the oncology community, even though the world has progressed in a long way since then. So the definition was that patients had to have a compelling rare cancer. We used the Comprehensive Cancer Panel at Peter Mac, thanks to Stephen Fox. We had a tumour board, both discussions at Peter Mac, but then also a virtual tumour board where rare cancer clinicians with expertise discussed each case and patient follow-up. This was presented by Dr. Damien Key, the first author, who had an oral presentation accepted from abstract at ASCO this year. The incidence of tumours enrolled onto nominated by oncologists from around the country were exquisitely rare, and that was very important. So we had 100 cases, and the frequency of actionable genetic events matched to drugs was obviously our main aim, not whether patients were able to access the drugs, because that was clearly not feasible in a country where access to drugs was nigh impossible. So of our 100 cases, 56% of rare tumours had at least one actionable finding, and 27% of those were of high impact. Timing, you would want to have that to be earlier during the patient journey to maximise benefits. And in order to have drug access within Australia, we would need trials with pan-tumour cohorts and drug approvals of a similar nature. And both of those have now moved in a pleasingly appropriate direction. So at the moment, we are collecting and sharing data to build further evidence to facilitate drug approvals. So I just want to switch now to the way we have built further infrastructure. Some of you may know of Cartwheel, and I'm going to describe the WEHI Stafford Fox Rare Cancer Program, and also finally our Rare Cancer Portal, which is now active and underpins triaging of patients, particularly in regional areas, into the Australian Genomics Cancer Medicine Program, which was funded initially from the Garvan, but now nationwide by the Health Minister in 2019. So Cartwheel is a consumer-entered data base which is available online, and it allows rare cancer patients to indicate they wish to participate in research. And the idea is to help us understand rare tumours better, but also help patients access research. So this provides links with rare cancer consumer groups. It's globally accessible. The international participation has been approved by Melbourne Health HREC, but we have now this year gained approval by the Western IRB in Seattle, which is a global IRB. And so we now have dual oversight. There's an online questionnaire and online consent. So how does all of this fit together? So we have COSA Rare Cancer Group oversight of all of this, and WEHI Stafford Fox sits in the middle, and I'll show you how it intercalates with the Rare Cancer Portal. So this is a national study of rare cancer cases. Patients will be able to provide consent physically in each capital city as their sites come online, but we've been doing so much upgrading because particularly of the portal that has delayed the individual sites coming online. In the meantime, patients have been able to consent through the remote consent option. We have had a genetic counsellor all the way through who's available by email and phone. So patients have been able to consent from anywhere in Australia from home. So then once they've consented, their clinical data and their tissue, either their baseline FFPE or fresh tissue from the time of surgery or biopsy, can be couriered, and blood, of course, for germline DNA, cell-free DNA, and PBMCs can be couriered as well. We've initiated a REDCap rare cancer database, which I'll show you, and most importantly, local investigators can use streamlined ethics and governance for their own projects. So also locally at my site, we have genomics and bioinformatics fellows and a medical oncology fellow, Dr. Damien Key, who I mentioned earlier. We have a whole range of individual projects. We've accrued more than 300 patients, 366 tumours, and we have a range predominantly of rare gynae projects at the moment, but other non-gynae samples have been accrued. This database is generic, and we focus on treatment-free intervals as surrogates of response, so we don't have to collect actual imaging data. I just want to highlight one project, Rare Gynae Cases with Whole Genome Sequencing, and this changed practice in our site, so we now initiate whole genome sequencing at first recurrence for rare cancers and at diagnosis for super rare cancers. And I'm just going to show one example of that. So this case, which I'll describe, this patient has gone public with her story, so I'm showing you quite a lot of detail. She had a rare tumor type, uterine leiomyosarcoma, and we found a homozygous deletion of BRCA2. She was provided with PARP inhibitor, was well on that for about a year. When a pre-existing nodule progressed, we knew from her patient-derived xenograph that that was sensitive to cisplatin, but that's a drug that is not usually given. So she then went on to cisplatin because that's a safe combination and went back into PR. So she's absolutely delighted and will report the fact that platinum is almost certainly a safe combination with PARP inhibitor, as these patients don't normally receive that. A second case was also found to have an HRD signature. The whole genome sequencing failed because it's a very heterogeneous sample. So at Lawn Cancer Conference, which some of you may be familiar with, we had as an invited speaker, an international expert, and there are Sean and Serena looking at the circus plot on the mobile phone. And AstraZeneca was happy to accept that additional international advice and to provide olaparib for signature 3, and she's almost the only patient in the world on it for that reason. So I just want to talk about the portal. So the portal is an extension that we have been building for some years, but it's now active. So this is a flyer approved to be given to patients directly, and it will enable their doctor to receive online expert advice so that they can stay with their doctor and not have to doctor shop. Consent is via the WeHi Stafford Fox Rare Cancer Portal, and this is the advice that their doctor will receive. This will prevent patients having to go to Rare Cancers Australia to ask for additional support when they feel, particularly in the regions, that their doctor doesn't have knowledge about their rare cancer. And this again is one of the reasons that our consumers came to us and said they had a problem that they needed filled. And this was funded $4.5 million as part of the $45 million package for the AGCMC. So this was incredibly important, and we've been rolling it out through Tasmania and Victoria, just about to start now through New South Wales, and it will reach out through all of Australia. So triceps is the international linkage. We still are trying to fund this properly. I'm chair of the International Rare Cancer Initiative, which is the global rare cancer board. And we do need to solve the funding problems for this. So these are my acknowledgements. This is an ongoing work in progress. Everything changes every couple of months, and we have a lot of work to do. But thank you for your attention.
In the interest of time, I think we'll stop the session. If you could all join me in thanking our 4 excellent speakers who each gave us an inspiring glimpse, I think, in what the future will bring in the area of precision medicine. So thank you very much.